Today we are privileged to hear a talk by Professor Maria Santore of the Department of Polymer Science and Engineering. Professor Santore received her PhD from Princeton in 1989 and joined our faculty in 2001. She has received numerous awards and honors, including being named a Fellow of the American Association of the Advancement of Science, the American Chemical Society, and the American Physical Society. Professor Santore is a world leader in the field of soft material interfaces. Her research addresses the universal chemical physical principles that govern interfaces within diverse material classes. Through her work, Santore has advanced the understanding of complex fluids, suspensions of particles and molecules such as those in paints and coatings, surfactants, foods, blood, vaccine formulations, ultra-thin 2D materials and films, such as graphene, polymer bilayers, and the cell membrane, and fluid-solid interfaces in biomedical implants, bacterial adhesion biofilm formation, and the surfaces of microfluidic channels in chromatographic packings. Her research portfolio spans the full range of science and engineering scholarship, with some programs directed at new fundamental understanding and others prioritizing translation and technological advances. This afternoon, Professor Santore will present her lecture entitled From the Surfaces of Cells to Materials Innovation, Synthetic Systems that Mimic the Behaviors of Biological Cells.
First, let me say how honored I am to be recognized with this lectureship and how glad I am to be given the opportunity to tell the community about my enthusiasm for science and my interest in translating fundamental knowledge into applications and translational useful technologies. I also have to say just how blessed I feel to be here at UMass with colleagues that are so full of ideas and willing to share, and students that are so hardworking and enthusiastic and just love what they do. So before I get into the story about materials that behave like cells, I couldn't resist trying to first tell you about my research on a general level, because people always say, Maria, I don't understand what it is that you really do. And it always takes a while to sort of break it down. But before I even get into the what and the why of my research, I have to start with the who. The students that have worked in my lab over the years, both PhD track students, master's students, and postdocs, and many undergraduates who have lived the life that advances the science and translates this to technology. I am in touch with most of these people still. Some of them emailed me just yesterday. They really, once they joined the lab, they're part of my life and part of my family forever. And also, I want to thank my collaborators, the excellent scientists, both here at UMass and outside UMass around the world, and the students and postdocs in those labs, and the agencies and companies that have sponsored the research that you'll hear about in the next hour or so. All right, so what do I study? Well, I study surfaces on colloids and interfaces and in thin films. And so I guess I'll start with the story of what is a colloid. Colloids are tiny particles. If you were to Google colloid, you would probably get pictures of boring spheres over and over again. But in my lab, we have spheres and some unusual shaped particles shown here, synthesized very recently by an undergraduate in the lab. And the colloid can refer to the particles, or it can refer to a suspension of these particles that you'd be very familiar with. For instance, milk is a colloid. It's made of little protein particles and fat globules that are suspended in liquid. And also, shaving cream, for instance, is a colloid. It's made of tiny air bubbles with a soap liquid around those. And the cool thing about it is that shaving cream doesn't behave like air, and it doesn't behave like soapy liquid either. It has properties all unto itself. And so that's one of the reasons why we're interested in colloids. Another thing about colloids that you should know is that they're very small. How small are they? Well, if you start with a length scale that's about 1 meter, and you slice it up, and divide it out 1,000 times, you get a millimeter, right? So start out with 1 millimeter and slice that up 1,000 times, and you're down to a micron. And now you're getting in the size length scale of colloids. Colloids are about a micron or maybe a little less. If you take that micron and slice it down, you're down by 1,000-fold, down to a nanometer. And so the smallest colloids are 50 nanometers, so the biggest ones are a few microns. The wavelength of light is a couple hundred nanometers, or a fraction of a micron. And so that's the size of these particles. And the reason why size matters here is because you get very counterintuitive behavior when you have things that small. When you have a round object like this, well, gravity works on it. The force of gravity is the mass of the object times the gravitational acceleration, or the force of acceleration, inertia that I have to overcome, is force times the acceleration that I threw it. Okay, but colloids have very small mass, so those kinds of forces don't really matter as much as the force on the surface of the particle. Okay, before I tell you a little bit more about that, I'll mention that other colloids with which you're familiar include bacteria, like these Staph aureus bacteria that are a micron across and they're spherical, and under an electron microscope like this, it's very hard to distinguish them from silica spheres here from my lab of the same size. And another bacteria that you're familiar with is E. coli that's in your stomach, and some E. coli can also make you sick. E. coli tends to be about 2 to 3 microns long and a little bit less than a micron across, and it's shaped kind of like some of these rods that we synthesize in my lab. So you might even imagine that bacteria are living colloids, and we can study both the physical behavior of bacteria and compare them to some of these synthetic particles that we make. Okay, so I made the point that things this small tend not to be governed by classical forces, but are governed by the chemistry that's on their surfaces. And that's why I'm interested in interfaces. Some of the surface chemistry that we take advantage of has to do with groups that put charge on surfaces. Most surfaces are already negatively charged, and we can take advantage of that. Other times what we do is we put polymers on surfaces. Polymers are very large molecules. They tend to be long strings or branchy strings. And when you put these kinds of molecules on a surface, you usually cause the surfaces not to stick together and even to slide over each other in a lubricating kind of way. Well, so already we're biomimetic. We have particles that look like bacteria, and the surfaces of these particles that we can make also look like bacteria. So here's an electron micrograph of subtlest bacteria, and you can see here the peptidoglycan brush that comes naturally on the surface of that bacteria that resembles the kinds of polymers that we put on the surfaces of synthetic particles. And here is an electron micrograph of the cells that line blood vessels, and especially the capillaries display this glycocalyx, which is a kind of naturally occurring polymer brush that lubricates the blood cells that have to flow through these tight spaces. And so when we put polymers on surfaces, it looks like the surface of cells and can do some of the jobs that you find already in nature. Okay, and then the third sub-area that I study are thin films. Graphene and their interaction of graphene with proteins is one of the subjects that we've studied. Graphene is of interest because it's an atomic interconnected layer of carbon that has interesting electrical properties, for instance, for sensing applications. And I'll talk more today also about another kind of thin film, that which makes up the cell membrane. Cell membrane is made of a bilayer of molecules that orients to form sort of an elastic sheet, and it's only 4 nanometers thick. So remember the length scales involved here. A millimeter is 1,000th of a meter, a micron is 1,000th of that, and a nanometer is 1,000,000,000th of a meter. So we're talking about things that are very small but very important. And my lab is interested in how these materials behave in water, how they respond to force and flow, and especially their weak reversible interactions. Oops, let's go back. All right, we're interested in these materials because they show up in so many different places. So they show up in paint and adhesive formulations. Believe it or not, I have actually painted a United airplane, but not this one that has the paint peeling off. I helped paint a 767 to evaluate the properties of the paint. I've been involved in projects involving fabric coatings and paper making. My lab has worked on hernia repair mesh. We've developed a coating for blood collection tubes as a collaboration with Becton Dickinson. All these crazy things that seem unrelated to each other all have these principles of colloids and surfaces. I'll be talking today about some of our interests in making materials for cell and bacteria manipulation. So we've also, in addition to helping companies, tried to translate some of our fundamental findings to new technologies. And I'll also tell you a story about fun we've had with materials that mimic white blood cells. Okay. All right. So when you have two surfaces or two particles that have surface charge on them, they tend to repel each other, just like two magnets that you bring together, that you bring the north poles together or this pair of south poles together. You have to force them together, and you put work into bringing them together. And it's almost like loading a spring. If you let go, they pop apart. And so the negative charge that's on the surface of many colloidal particles or naturally occurring surfaces behaves this way. And we can think about that, quantify it, by thinking about the energy in the system when we squeeze the surfaces together. We load it up, and that increases the energy of the system made up of the pair of particles or the pair of surfaces. Now, I can do chemistry, let's say, uniformly on that surface, and it would change the nature of the interaction. It may be that I can make this surface somewhat attractive towards the particles that I'm interested in studying. And in that case, when the surface is far apart from the particle, it may feel like it's pulled together. And so that would be represented here as an attraction because the system is doing work on me. I would have to pull on it to hold them apart. But it also may be that this attraction only happens at some range, and if I worked harder, I would have to put some energy in or squeeze to push those surfaces together. So it's possible to have attractions at some length scales as a result of chemistry and repulsions at other length scales as a result of chemistry. And when that happens, there's a minimum in the interaction, and the surfaces or the particles like to sit at some happy separation apart from each other. And that could be, for instance, the basis for lubrication. Once they're in that minimum, they could slide over each other, and you would have to do work and apply force to move things around. Now, my lab discovered that while you can do uniform surface chemistry to make something like this happen, you get a very different result if you take the same chemistry and the same amount of surface chemistry and just put it on the surface differently. If you cluster it into tiny lumps instead of smearing it uniformly off the surface. In that case, you quite often get an interaction that's far more attractive than the situation where the interactions are smeared out uniformly. And that attraction might still give you a minimum, but it means that the interfaces like to hang out more often closer together than further apart. Okay, and we've taken advantage of this unusual finding to create some new ideas about technology. So we've made series of surfaces that have length scales in them. The obvious length scale is the size of the chemical feature itself. It also may include the average spacing between randomly positioned objects on a surface. And it also has some other length scales having to do with the random arrangement, the size of an effective hotspot on the surface, if I were to come and stick to that surface. And we've made surfaces that look like this with nanoparticles from Rotello's lab in chemistry. We've used polymers to make surfaces that have this kind of heterogeneity. We've taken nanoparticles and put the polymers in between to create heterogeneous surfaces. We've put the polymers on the particles, and we can put polymers in between that. So there's all kinds of things that we can put into these systems depending on what we want to do. Now, one of the cool things that these surfaces do is to adhere targets in a very weird selective way. And I'll try to explain why that is. If we start backwards and think about the features that we're ultimately going to put on a collecting surface, but think about how they might interact with colloid particles in solution, we can come up with particles that have the same kinds of surface chemistries that are different sizes and shapes, and it wouldn't matter. These little stickers that we're ultimately going to put on our collector would be equally attracted to these different particles in solution. So we say the adhesion is nonspecific in a case like this. Now, if we take these stickers and distribute them on a collecting surface and consider how particles from suspension might interact with the surface as a whole, we get a different result. And we get this selective result. And you can see why here. This big particle, if it's going to try to stick to the surface and is going to be repelled from the blue part, might try to sit down on a hot spot, maybe made of two stickers. But when it tries to do that, it will also have interactions with the rest of the surface surrounding that little hot spot. Okay, now, if you have a smaller particle or a more sharply curved particle sitting on a same pair of stickers or a hot spot of the same magnitude and same level of attraction, then it doesn't feel as much pushing because it doesn't have as much aerial contact with the rest of the surface around it. And so the relative amounts of attractions versus repulsions that are felt for these different shaped objects plays out differently. And we can have situations where big or more gradually curved objects are not as adhesive on the same surface as exposed to the smaller particles. And from this, we can imagine situations where objects of different shape have different amounts of adhesion to the surface, not based on their weight, not based on their overall size, but simply based on their curvature. Okay, and so what we did was we developed surfaces that look like this, and we exposed them to mixtures of particles. We have here a mixture of particles that's made of half and 1 micron silica spheres, and when we expose it to a control surface that's sticky everywhere, you can see both the big and the little particles sticking to this surface. However, an engineered surface exposed to the same mixture of particles only adheres the little ones, and it is a very sharp selectivity. We can quantify that selectivity by counting the number of particles on the surface. In this case, it's almost perfect selectivity. There's one little feature here which may involve a big particle that looks to be aggregated, though, against some other particles. There are no big particles all by themselves here. And so based on the fact we only have about 1,000 particles on this surface, we can claim a selectivity of 2,000. So we can put numbers on this. So in applications where it's important to purify particles, this is a really fast and easy way to do it. But perhaps more valuable still is to take these ideas into the biological arena, which is what we tried to do, and we developed surfaces that would do the same thing for cells. Here's an example using Staph aureus and E. coli, the 2 bacteria that I introduced as being colloid-like early on, and we could take a mixture of them. I've got them colored based on the Gram staining. It turns out that Staph aureus tend to take on a purplish stain, and the E. coli tend to selectively pick up a pink stain, so I've drawn that in with PowerPoint ahead of time. But we take a mixture, 50-50, of the 2 kinds of cells and flow it over one of our surfaces, and the selective surface will take only the E. coli out of solution, but the control surface, of course, takes both. So we have a surface here based on curvature that will collect E. coli cells and not Staph aureus cells, so we can selectively grab onto different classes of bacteria. We've also taken this idea in the direction of mammalian cells through a collaboration with Kathleen Arcaro in the Veterinary and Animal Science Department, and she's very interested in breast cancer and assessing cancer risk, looking at a milk that's obtained from lactating women shortly after they have children. And so that milk contains cells that have sloughed off the inside of the breast ducts, and it's mixed. Those cells are mixed with the milk itself, which is proteins and fats, and also contains white blood cells. So it's a very complicated mixture, and we have surfaces that can grab the breast cells out of that complicated mixture. But I'm showing you something here that's even more amazing that we don't quite understand. And that is, we have a surface that can distinguish 2 related cell lines. This one here will be killed by the cancer drug tamoxifen, but this one is a tamoxifen-selected clone, and it's been dyed green, so it is a more aggressive kind of breast cancer cell that will not be treated by tamoxifen. And so we have 2 different kinds of cells that are similar except in their susceptibility to these cancer drugs. And what you're seeing here then is a series of micrographs. This is a fluorescent channel of the same sample, and this is with all the lights on so you can see all the cells. So all the cells are over here, and the ones that glow green are the tamoxifen-resistant cells on the fluorescent channel. And it's hard to see what's what, so we light them up by PowerPoint, and then we take this pattern of dots and move it over to the other side. And so what you can see here is that more than half the cells, a substantial number of them, about 70%, are the ones that are tamoxifen selective. We only have a little bit of cells that are the more susceptible, less aggressive cells. And you compare that to a control surface here on the fluorescent side. These are the tamoxifen selected ones that adhere from a 50-50 mixture, and you move those dots over, and you can see really about the same proportion of cells adhered on the control surface in proportion that was what was present in the bulk solution. Okay, so these surfaces can adhesively select for different cells based on the features of their surface, some of which we don't understand because the biologists don't yet understand, but these differences exist. All right, and the final point I want to make about this is that we're doing this based on materials and physical principle. We're not relying on biomolecular recognition. So a lot of these kinds of tasks can already be done, but they're done using antibodies. And those antibodies are very expensive, and they're fragile proteins. They can cost upwards of $300 per 100 microgram of material that you would need to make an adhesive surface. And the materials that we're using for this are polycations that are on the order, if you buy them fresh and in small quantities, on the order of $100 for several grams. So it's a substantial savings cost-wise, much more economical to make and perhaps to use. All right, so that's an example of a fundamental idea that we observed in the lab and how we translated it all the way up to something that might be useful in a healthcare application. That's sort of mature. We also have some fun in the lab, and we're not sure yet what it would be good for. And I'll tell you that story next. This goes all the way back to when I was in grade school. One of my favorite things to watch was the science videos, and one of my favorite things to watch when it came to science videos were pictures of cells doing stuff. And I can tell you the 4th grade at Pine Ridge Elementary School would uniformly agree that amoebas were the coolest cells around because of the way they moved, the way they sidled up to things and then just sort of engulfed them and gobbled them up. And that was it. So this is just really cool stuff. Everyone seems to recognize that it's cool. I don't have to convince you of it. So there's the cell. It's big. And the target here is about 1/2 micron in size. Now, from YouTube is an example of a white blood cell here. It's stuck to a surface, and it's smelling somehow the fact that there are some bacteria here. And it's doing basically the same thing. It's engulfing these bacterial particles, but it's stuck to the surface. So the process is kind of tangled up by the adhesion of the cell to a surface and by the adhesion of a bacteria to the surface. So it still happens. Perhaps more cool still is this experiment that came out in 2006 or 2007 by a group at UC Davis. And I'll have to tell you what to look for because I'm going to play this once. This is a white blood cell. It's held in a micropipette. It's about 10 microns across. And it's going to eat this glass bead here, which is about 4 microns across. And the glass bead is coated with something that would be tasty to the neutrophil that says, I'm a bad guy, I'm a foreign agent, you need to get rid of me. And it's sped up. The whole process takes about 30 seconds. Believe it or not. And what you're going to look for here is that after contact is made, there's a little bit of a time lag, and then the engulfment happens. Then it will repeat, but the bead will be bigger. There'll be a 6 micron bead next, and I'll warn you that the time scale there is going to be about a minute, but you'll still see the time lag. And then it'll repeat again with even bigger bead that's about 11 microns, and it takes 20 minutes as the cell struggles to engulf it. Okay, so here it goes. Blammo. 30 seconds start to finish to engulf this bead. That's 4 microns. Here's a 6 micron bead, a little bit of a lag time. It's tasting it. Oh, yeah, it's good to eat. Down it goes, down the hatch. Okay, now the 11 micron cell. This is why you don't try to put big things in your mouth. It takes 20 minutes, it struggles, it never manages to do it because the bead is just too big and the cell can't manage to wrap itself around. So the features to look at are the apparent lag time and the fact that size is important again here. The process of engulfment by a cell involves the adhesion of surface receptors between the cell and the target, processes that then happen at the surface of the membrane, and then the polymers or cytoplasm that are inside the cell rearrange and the cell flows out and around its target. Okay, so that's how it works. The other thing I should point out, sort of related to phagocytosis, is this other engulfment process called endocytosis, which is especially relevant because it's how viruses infect cells. So a virus is a very small particle compared to the bacteria that are being attacked through phagocytosis. A virus particle here is about 50 nanometers, and so there are no movies for this. This is a series of electron micrographs of different stages of viruses being tricking the cell to take them up. the virus adheres to the surface of the cell, and then the membrane wraps spontaneously around the virus without substantial flow of the cytoplasm. And so that process is called endocytosis. Okay, so depending on the size of the target, the cytoplasm may or may not be involved, but the membrane certainly is. And there are sort of a range of different processes that take place depending on the size of the target, from the phagocytosis, where there's a substantial rearrangement of the cell shape, to endocytosis, where the process is really driven at the level of the membrane. And then there are some things in between. Okay, so if we want to make an artificial amoeba or an artificial white cell, I think the first question to ask is, what is the simplest system we can build that will do the job from a materials perspective? Can we do this without a cytoplasm driving the process, without these metabolic processes that do it? Will we be sensitive to the same size constraints? Will the process happen on the same time scales? And then this paper that came out in 2006, after we had completed our experiments, but just as we were writing up our work, shows the range of tensions in the micropipette that were holding the white blood cell, all the way up to these units called millinewtons per meter. That's sort of a unit of force in the tension of the surface of the cell. Okay. So when I think about engulfment processes, if I want to start simple, I will start with the idea of how a liquid drop wets a surface. That's a process on the materials side that looks kind of like engulfment, and the shape of the water drop and the apparent extent of engulfment depends on a couple of things. It depends on the surface tension of the drop itself. And so I think quite a few of you recognize that water has a substantial surface tension due to the arrangement of molecules right at the skin of the water. It's a rather stretchy material. Something like gasoline has a much lower surface tension. It tends to flow out over surfaces more easily. So there's sort of a surfacey, stretchy kind of process that goes on here, as well as in the biological case. But also involved are adhesion processes right here between the droplet and the surface that it's spreading over. We can begin to think about this, if we dare, in terms of a force balance that takes place at this corner of the drop where spreading is really happening. We can think about the surface of the liquid pulling back, the contributions of the liquid-solid interactions, and then what the energy is of the bare solid as well. So that's one way to think about it. And these numbers that I showed you, they're big compared to the 1 millinewton per meter that was measured and reported for the white blood cell that was having lunch. Okay. Now the cell membrane that's involved in endocytosis and somewhat also in phagocytosis, we have to think about how that is made as well. It's made of an array of molecules called phospholipids that have fatty tails that don't want to be in water, and a polar head group which is happy to be in water. And so because these fatty tails don't want to be in water, they assemble into an arrangement where they're shielded and not really wet. And it's only the polar head groups that face the outside of the cell and experience the water, and polar head groups on the other side that face the water on the interior of a cell. And so that's what a cell membrane looks like. In the polymers community, one can obtain polymers that have two parts to them, very much like these phospholipids, but only bigger, higher in molecular weight, where part of the polymer might be water-loving and another part of the polymer might be water-hating. And these kinds of molecules also assemble into these bilayer lamellar structures. And what you notice is that the surface of this bilayer also has the same kind of fuzz on it that I argued earlier was valuable in a biological setting in terms of being like a glycocalyx or being like a peptidoglycan layer that would be on the surface of bacteria. That's already integrated into the surface nature of these copolymer membranes. So what we're going to do is we're going to take the copolymer membranes and make them, through a strange process called electroforming, into sacs that are very much like the sac of membrane that's around a living cell. Okay, and those sacs are called vesicles. There's water on the inside and water on the outside. And it's what you see happening here. They're growing on an electrode and they're shaking because the electrode has a frequency that's being put through it. And the thickness of these bilayers here, the sac, the membrane skin, is only 4 nanometers thick. Now, you can't see 4 nanometers in an optical microscope. We've played some tricks with the polarization, so the light reflects off the surface of the vesicle, and so you can see where the edge of it is, and it looks like you're seeing the membrane itself, but it's like a soap bubble. A soap bubble is also thinner quite often than the wavelength of light, and you really see the reflections in the light interference as well. So this is how we make these vesicles. The membrane is only 4 nanometers thick, but the vesicle itself is like a cell, is several microns across, 10 microns or 20 microns across. Okay. And we can pick up these vesicles in micropipettes and pull on them, much as was done with the white blood cell that you saw. Here we're pulling back on it. It's like aspirating a balloon into a straw, and we're pulling on it, and then the further we pull, the more we stretch the membrane, and the harder we have to pull, and then it breaks. And we can analyze based on how hard we pulled and how much the membrane deformed, the stress versus the strain, and report the stretchiness or modulus of these membranes. And they're high. They're higher than the surface tension of water, but that's not quite the whole story of the vesicles. So we take these vesicles then in our lab, and we do an experiment that's very much like what was reported for the white blood cells eating the glass beads. You can see here these two little rods coming together, and at the tip of them are the glass micropipettes. And each micropipette holds a vesicle. We're not going to use a glass bead in this experiment. The left vesicle is being aspirated with a substantial suction, so the part that protrudes out of the micropipette is always round. And the part on this side, it starts out round, but because it's at lower tension, as the engulfment proceeds, the shape changes. Okay. And then as the engulfment proceeds, we measure things very quantitatively. We can quantify the contact angle, the contact area, the detailed shape of the vesicle, and the protrusion lengths and the area changes. Part of our analysis has to do with an equation here that looks very much like what was established for spreading of water drops on surfaces. So we call a little bit upon this kind of physics when we do the wetting analysis and the spreading analysis here. And our early experiments here, we took these two brushy surfaces and vesicles, and we put them together, and we poked them at each other, and this is sped up by about 10 times. But you can see really there's no adhesion. They're sliding over each other as evidence for the lubricating nature of the polymer brush that's on the surface of the membrane. They slide right over each other. You can try to stick them together, but they won't. Okay. Now, if we modify the surfaces of these membranes with some chemistry that makes them sticky, okay, then we can have a situation that looks very much like the neutrophil eating a bead. And this video that I'll show you next is in real time. You're going to see 24 seconds of video. And remember to watch for the lag time, just like in the white blood cell case. It takes a little while before the surfaces really come together, and then you see a spreading and engulfment process happening. It doesn't go all the way to closing off like a white blood cell, because we don't quite have enough membrane in this side to eat around another object that's so big, but we definitely move in that direction. Oops. Okay, and we've, for all these different video frames, we analyze the details of this, so it's quantitative. We have a lot of fun with the movies, but we ultimately bring it home in terms of explaining the forces that are taking place. And so we take still frames, and we measure things like contact angles and the size of the contact region. We've identified exactly how long that lag time is, and then the rather fast engulfment process that follows as the adhesion is established, and then what follows as the vesicle is pulled out of the micropipette here. Okay. And we've done this for different tensions in this micropipette. So we can impose a constant backward force in the low tension pipette that's holding the effective neutrophil that we've made. And we can pull back so hard that we get numbers from about 0.02, 0.7, and 1 millinewton per meter. And so I'd like to remind you that those were basically the range of numbers that you saw for the neutrophil that was eating the glass bead, quantitatively. Okay, and the time scales involved to do the engulfment process are also very similar. Okay, so we're pulling back on this, but we've always observed at the end of the run, the vesicle always escaped the pipette. It just would never be happy being held back, and we wondered whether we could stop that from happening in this case. It's sort of, like this. The vesicle wants to go where it will, and we're pulling back as hard as we can, and it's a tug of war to see if we can manipulate the material system with the micropipette. And in this case, I think the cat pulling back is more strong than we've been managing to do with the micropipette. So the next experiment we did is once we saw the adhesion start to happen, we then ramped up the suction pressure and tried to pull back and say, hey, wait a hold it, we want you to stop engulfing. Will you please stop? Right? So that's what this next experiment is. And it's very hard to see the way the lighting is here, but I'll also show you some still frames. You can see the projection here. The adhesion is happening. We're pulling back. We're pulling back. We're pulling back. And then it's still there, but it's going to break. It's gone. Okay, so we had to pull back so hard, but we couldn't tear it apart, and we couldn't even stop it from advancing. The membrane broke. We went all the way up to the lysis condition of the membrane. Okay, so here's the quantitative, and we can enhance the stills so that you can see them a little bit better. Once we got past the lag time and saw the adhesion happening, we increased the force, the suction force back the other way, and we went all the way up to the lysis condition of the membrane. And we couldn't stop the adhesion from happening. So we've made these very aggressive engulfing scavengers. And then, the happy story is we can make them either way. We could also re-engineer the system so that we have a reversible adhesion as well. So in this one, we can pull it back, but you'll see it also pull out. We let go at the end. So we pull it back. We can control the reversibility of it in this case, or let it go. So depending on what we're doing on the surface, we can make a system that advances the adhesion right up to sacrificing itself as a membrane, or can adhere reversibly. So that's one fun little story that we've had making an artificial white cell that can do phagocytosis. I thought I'd tell you another story about where we are now, thinking about other things that immune cells do. There are dendritic cells, which are a special kind of white cell as part of your immune system, that when they encounter an enemy, like a virus, they eat it up. And then like a child, they smear the pieces all over them. And then they go and have a conversation with another immune cell to communicate what it was that they just ate. See, Mom, it was delicious. And so there's communication between these two cells at this point, and this immune synapse is something that's really fascinating. There's a pattern of the arrangement of molecules and receptors at that immune synapse where the cells are talking to each other. And so this idea that patterns of arrangements of chemistry on the surfaces of cells is related to their communication has motivated us to try to understand what we can do to make cell-like membranes that could ultimately adhere cells and ultimately enable people to communicate with cells. And so the students in my lab start with phospholipid membranes, and then they add this particular molecule, and if you notice, it doesn't have any double bonds in it. If you're a chemist, it's a trans molecule. So it is the trans fat kind of molecule that you're not supposed to eat because it's bad for you. It solidifies in the membrane, and it orders into something fascinating. It's a two-dimensional crystal. So the membrane, remember, is only 4 nanometers thick. The molecules move around in it, so that qualifies it as fluid. And then these guys here can solidify and make solid domains. So it's like having a cold glass of water or iced tea that you've added sugar to. Some of the sugar dissolves in the liquid, but the rest of the sugar might not go in because it's so cold, and you have sugar crystals on the bottom of your glass. So you have a sugar solution in equilibrium with sugar crystals. The same idea here, but it's 2 dimensions. It's only 4 nanometers thick. So it brings all kinds of other physics into the problem that we'd be interested in understanding. Okay, the other thing that's even more complicating about this is this particular sort of fatty lipid doesn't just order into this tilty kind of domain. It can also order into a zigzag kind of structure. It's really quite beautiful here. This image is from AFM. So there are two different kinds of crystals that this kind of molecule can make. And if you're not comfortable with that idea, how many people like to read science fiction? A couple. How many of you have read Kurt Vonnegut? Okay, not as many. The rest of you should. The book that I think of is Cat's Cradle, right? Because in Cat's Cradle there was a form of water that solidified called ice 9, and it was different structure from regular ice, and it was a problem, and it advanced the plot of the book, and I won't quite tell you what happened there. But so there's an example of two different kinds of solid crystals. Okay, well, what we realized about our different kinds of solid two-dimensional crystals is that they had different numbers of molecules per unit area, and as a result would be sensitive to any tension that we imposed with those micropipettes. And so if we're at high tension, the system should prefer, if it needs to solidify, into choosing the crystal form that is less dense. And if we're at low tension, then it would be allowed to form the more dense crystal phase. Okay. And indeed, that's what we showed, is that depending on the tension that we imposed, in this case osmotically, we would get one or the other of the crystalline arrangements in this 2D membrane. But more cool still is that one kind of molecular packing of the molecules gives rise to vastly different morphologies and different connectivities than the other. So we can pattern these membranes based on having one kind or another kind of crystalline arrangement in the solid phase. Okay. Furthermore, I thought I'd tighten up the analogy with the Kurt Vonnegut story. One of the things that happens is that there are these little seeds of ice 9 that if they touch regular water, they'll nucleate and cause the rest of the water to freeze out into this bad ice 9 structure. So the idea of nucleation and growth present in the Kurt Vonnegut book is not science fiction, it's science fact, it's real physics, and it also happens in 2D membranes. So if we have little seeds of this particular this is the zigzag phase, and we allow it, it will grow. Okay, so you can have seeds that grow into larger domains on the surface of a membrane, and that allows us to do quite a lot in controlling the pattern depending on how many seeds we put in. If we put in just a few seeds, then we get a pattern of big dots. If we put in lots of seeds, then we get smaller dots in the end when the system equilibrates. You know, I like to say that one of my favorite things about working with UMass is the students, when they come to me with beautiful data, it just makes my day. It doesn't make my day, it makes my entire week. And so one of my new students came on the vesicle project and showed me these images at group meeting probably about 2 weeks ago, and I couldn't help myself. I had to include them, even though they're really done with only a 40x objective on a cheap optical microscope. You know, I couldn't help but looking closely and then more closely. And they're just gorgeous. They have such ordering to them. They're all uniform in size. You know, look closer still and imagine what we might do once we get over onto the facilities and aisles. Okay, so we can control these patterns of spots. We can make beautiful arrays of stripes and of patches depending on the thermal history and the composition that we put into the membrane, and through control of tension as well. Some of the things that we're doing moving forward into are pulling tethers on the membrane so that we can make objects that look like swimmers and could perhaps have interesting hydrodynamic interactions and flow. We can't quite make those things flap around yet and swim, but boy, that would be fantastic to do. And so that's where we are with that project now. All right, so we have a lot of fun in the lab. I love working with my students. I'm so happy to be here and share some of these ideas with you. We like to play. We're always fundamental, and a lot of these things have some really cool uses for the future. Thank you so much.
Professor Santore, thank you for that wonderful lecture, and more importantly for all the wonderful work all these years that has gone into the beautiful results that you showed today. So it's my pleasure to present you with the Chancellor's Medal, which is the highest honor that's awarded to any faculty member on the UMass Amherst campus.
Thank you so much. Thank you.